The overall goal of this protocol is to use digital analysis tools to quickly make accurate counts of cells in a suspension or on a migration and invasion assay membrane.
This protocol can help researchers quantify cell number required for common techniques and in vitro cell motility assays.
The main advantage of this method is that it provides fast and accurate cell counts by including multiple filters and analysis tools.
To begin, set the microscope illumination to maximum, switch to the 4x objective, and ensure that the phase contrast filters are in use. Then, within the microscope software, set the image capture settings to their default values. Now place a standard hemocytometer onto the microscope stage and capture an image for the image volume calibration step. Adjust the exposure time as needed. In ImageJ, under the cell concentration calculator, open the image and click on the Get Image Dimension button. This action fills in the image width and height text boxes with the image resolution in pixels. Next, select the Straight Line tool and draw a straight line across the entire length of the hemocytometer's primary p-square by clicking and dragging the cursor. Then, push the M key to display the results window. Now, type the value from the length column into the p-square length text box in the cell concentration calculator. Then, click on the Calculate Image Volume button to output the image volume into the image volume text box.